So Sting, yep, I think I remember that. I don't want to talk about it too much early, but I have to say that like it has been 100 years since I read a paper where the first figure was led by a gel.
Oh
Like
no.
I had a near panic attack when I opened this paper.
This is Audiomunity, a podcast about our body's never-ending fight with the outside world. Hi everyone, this is Audiomunity, episode 37, recorded on March 25th, 2025. I'm Kevin Bonham, coming from Boston, Massachusetts, and joining me, as usual, is Matt Woodruff.
Hey, everybody.
Down in Atlanta. And Kate Franz from Bay Area, California.
Hey, folks.
She's back, not blowing us off this time.
Oh, gosh, I was so sick. I was like telling people, after like the first year and a half of daycare, you don't get sick anymore. And then I was like, oh, my husband is dying of a terrible cold. I got some horrible flu-like illness. It's like, oh, I just don't register it anymore. Okay, cool.
No, this year was particularly bad. I mean, for a while I thought it was just like anecdotal or just like our family, but it really seems like from all the epidemiological evidence and from everybody I talked to, this winter was freaking rough. Did you have any of the two bucket disease? I mean, we had like two bouts of what seemed like noro come through. I had some like viral conjunctivitis a couple weeks ago. Like it's been bad.
Yeah, as much as I want to talk about two bucket disease on the internet, no, I did not have two bucket disease. Thank
Good
you
for
for your concern. Yeah, but we've just been, I think we've been luckier than most, but it still hit us. And yeah, but now we're recovered. Spring has sprung. It was 74 degrees yesterday. I was dying of heat exhaustion.
That swung fast, Kate. It swung really quick. We all the dogwoods popped this morning here in Atlanta. So the trees are all white. The azaleas haven't come out yet. So that's the true spring thing that happens in Atlanta. The entire city turns into azaleas that are flowering, which is pretty cool. But it's definitely here. It's happening.
I saw my first daffodils. We have a single crocus in our yard. So we're a little bit behind Atlanta, I think. But yeah, it's definitely warming up. I think I still have, I think I have a cold right now, and I thought I was going to lose my voice, but thankfully I'm here. I am mostly going to shut up for this episode, though, because the paper that we're going to read is complicated. So I think we should skip all of the politics that Matt and I have been doing over the past few weeks, and just get straight into this paper. So Kate's going to talk about it. It's called OAS cross-activates RNaseL intracellularly through cell-to-cell transfer of 2,5A to spread innate immunity. Very, very cell press title. This is in the journal Immunity, and the first author is Wanwan Hui, and the last author is Nan Yan from UT Southwest. And yeah, I mean, we can just get straight into it, but I just want to say, on the record, I remember Kate basically having this hypothesis 12 years ago that there were going to be second messengers that were passed through gap junctions between cells. And it is wild to me that now, over a decade later, there's actually an immunity paper about it. So, Kate, take it away.
So really, Kate is the first author of this paper, actually.
Absolutely not. Absolutely not. Nope. Yeah, I worked on a similar idea for a short period of time in grad school, looking, and it sort of, for myself, and I think probably for this paper as well, starts with trying to understand how is intracellular DNA sensed, and what is the intracellular DNA sensor in cells. And I will say that when I saw this paper, I was like, okay, this paper basically lays out like the exact experiments I would expect to see, that I would want to see. But why am I interested in this paper other than like having, had some thoughts similar in the past? And I think I want to talk about cancer vaccines. I think we'll get there eventually, and maybe this will be a part one of why should we care about cancer vaccines again. But I do think
it a vaccine if you want to get it funded. In
immunization.
Yeah.
But I had this realization last night, and I was thinking, is there enough time to like put some background on cancer vaccines? And then I was like, okay, no, we're going like too far afield. I think we will end at cancer vaccine, just like a little signpost for where this journey might end. But as maybe a teaser for a next episode, I did Google something very general about a cancer vaccine and saw a paper written in saw an article written earlier this year that said, the headline was something like cancer vaccines finally show promise. And then I see an article directly underneath it from 1989. Cancer vaccines finally show promise. And so yeah. I will say, personally, I do think that cancer vaccines are finally showing promise. Cool. But, we have been fooled before.
Well, it's funny you say that, because when I looked at this, and we're getting a little bit ahead, but that the cancer figure in this paper seems totally out of left field for me. So I will be interested to hear your perspective on that. Because when I saw that figure, I was like, what is this doing here? Like, that doesn't belong.
Yeah.
But yeah.
I think the place to start with this paper is probably background on DNA sensing, which is very funny because this is an RNA sensing paper. But it is, I was going to say it's like a spiritual successor, but it's actually a direct scientific successor of an earlier paper. So in the early 2010s into the mid 2015ish or so, there was a lot of intense intense research occurring over trying to identify intracellular DNA sensors. Specifically, I want to say that when we talk about sensor in this sense, what we're meaning is a protein within a cell that binds to a ligand and then transduces a signal downstream, and in this case activates transcription, activates interferons in particular. So we're looking at innate immune signaling. And so in this case, really what everyone was thinking about was the transcriptional activator that will coordinate the type 1 interferon response. So similar to RNA sensing, we have ring IMDA5 binding RNA directly engaging with the sensor MAVs to induce transcriptional activation that ultimately will activate type 1 interferons. The field was looking for the same pathway, the cognate pathway for DNA. And so there are a lot of DNA sensors that were identified. I think it's really difficult because when you start, because DNA, the form of DNA that is immunogenic is the DNA that is in your cells. Your cells are filled with DNA that is potentially immunogenic if it were to enter the cytosol. I think when you start messing with particular proteins, we start to get into more fundamental cell biology, and that can lead to these very difficult to interpret phenotypes. For example, you might be able to knock out our protein, you break the cell in such a way that it's no longer responsive to DNA, But just because when you knock out this sensor or knock out this protein and you lose your response to DNA being presented in like this immunogenic fashion, doesn't mean that that's a sensor. That's why I also at the top is like, you need to show that you can transduce a signal down to a second protein, or, I mean, it could be its own protein, right? But spoiler, it wasn't. So there's all this research that's going on in 2012, 2013. I don't remember the exact date. I think it was the end of 2012, maybe beginning of 2013. James Chen's, okay. So one piece that we didn't know about this pathway, sorry, I jumped ahead of myself just one step, is people found the transcriptional activator first. So they found the protein that coordinates the transcription factors first. And in the case of DNA sensing, that was this protein called STING. So Glenn Barber and a couple other people found and characterized this protein. And then once we found STING, we were looking for the ligand or the upstream sensor. It was very clear that STING itself couldn't bind DNA. You couldn't pull down STING if you transfected in DNA and then pulled the DNA out of that lysate. But what was found from Russell Vance was that bacterial cyclic dinucleotides could bind and activate STING to some low amount.
Kevin, it seems like you have magically moved from one recording location to another.
Yes. I am in this wonderful position where I am a computational researcher that cannot connect to the internet at this hospital. So
that makes all the sense in the world to me.
It really does.
If you said the internet was fast and freely available, I would say, you got to run, Kevin.
Yeah.
They have far too much faith in you.
No, it's fun. So there's one network that I can connect to that is very fast, but basically everything is blocked. There is another internet that I can connect to that is particularly terrible, especially in my office. It's like the guest Wi-Fi. That's what I have to do most of my work on. So yeah, super cool.
Have you thought about like an Ethernet cable?
Well, sure. That's the way to connect to the fast internet.
No.
But it's the hospital internet, which is like totally locked down because physicians that connect, you can only, like, I can't even connect to that Ethernet with my laptop, my normal laptop. I have to use a hospital provided one, which I can't install any software on.
Yes.
Again, as a computational researcher, that makes a lot of sense. So, yeah.
I actually am familiar with this problem. Yeah.
It's horseshit, but it's where we're at. So now I'm in the conference room. Better guest Wi-Fi. I might be interrupted, but we'll see.
Conferencing.
Carry on.
That's all right.
Okay.
That's all right. So we were talking about Sting and all the things that Sting can do.
I'm gonna just give a little bit of a. I don't want to start from the top, but I'll just give like a quick bullet point and then I'll continue on. So, race for intracellular DNA pathway that activates type 1 interferon. That's important because it's going to help activate the rest of the immune system. It's not just going to be, antiviral effectors. Glenn Barber and others find STING. They show that it can activate type 1. Russell Vance shows that there is a ligand for STING found in bacteria, cyclic dinucleotides, which are signaling molecules within bacteria. But it was unknown what the endogenous sensor and ligand was. Around 2012, 2013, a grad student and a postdoc from James Chen's group dual publish in Science that they have found both the sensor for intracellular DNA and that it makes a small ligand that activates STING. So that sensor was cGAS and the small ligand is cGAMP. And it also is a cyclic dinucleotide. So, we got some clues from bacteria. Turns out that same type of molecule was actually being made by a synthase in human cells as well. But it's the first metazoan cyclic dinucleotide that's identified, which is was very, very
And
interesting.
did you say what CGAMP and CGAS are? Like what those stand for?
Like
cyclic,
the words
cyclic
with the
GAMP,
letters.
right? Like the nucleotide,
Yeah.
like GTP, like chemically modified to basically like form a ring, right? And then CGAS is the cyclic GAMP synthase. Is that right?
Yeah, cyclic GMP AMP synthase. So
Synthase makes you make it.
Correct.
That's my
Yes.
contribution to this paper. Yeah.
Exactly.
Synthase is a protein that makes something.
So
Also, I had to just go back and remind myself what metazoan was. This is how far. This is how far you get down the rabbit hole where like basic high school biology becomes sort of like this vague thing that you once knew.
Fair.
Oh man, this is really funny because recently I've been thinking about all the things that I learned and then forgot. And man, Matt, you're making me feel real good.
Yeah, no. There's the whole base of a pyramid of biology that no longer is relevant to anything that I've thought about in like 15 years. So,
Yeah, but I feel like those neurons are still there in your brain somewhere.
Yeah.
Like that particular neural network, it just has lost its connections to the vast majority of other things. But I
Yeah.
feel like
No,
if you got a few little pings on that network, it would reactivate. You would come
Yeah,
back.
no,
I feel that way
and
about
actually,
flow cytometry plots.
yeah, and then actually I was just
Kevin.
about to say, yeah, I was chatting with Kate at the break there, and I mentioned that opening a paper where there was a gel as figure 1A felt like PTSD, like stepping to the edge of a cliff that I was going to jump off because, yep, all those neurons started firing, and they all said to me at once, "What are you doing?" And I thought to myself, I don't know. But all that is to say, I really like doing this podcast with you guys because this isn't a paper I would have ever read.
Yes.
So going back to the paper.
We have stunned Kate
No,
into
actually,
silence.
we haven't even
That-
started the paper.
That
We
is
haven't even
impressive.
started the paper.
I just want to say that's impressive.
Oh boy. So metazoan cyclic dinucleotide. Great.
Yep.
So ultimately, okay. So this came out to a lot of fanfare. I mean, because we won't do the papers for obvious PTSD reasons for 2/3 of this podcast, but the papers are stunning, beautiful. They're some of my favorite papers that I've ever seen. The biochemistry is incredible.
I remember them coming
and one
out
of
and us talking about that. I remember. That's
I
burned into my
I
brain.
cried. I cried when I read them because at that point there had been 15 DNA sensors that had been proposed, but the biochemistry was so tight. It explained why no one had ever seen it before. It had the full mechanism. It had an isolation and a characterization of CGAMP. It was just so great. So I did not cry because of the stress of it. It was just like, oh, this is beautiful. This is elegant. I completely see the pathway. This is it. We figured it out. And I think when you are in a field that has had so much back and forth and half phenotypes and almost there, to actually see that resolution that everyone immediately says, oh yeah, this is what it is just so exciting. So ultimately, it seems that this is a pathway that has a small molecule that is helping transfer the signal. And so this becomes very interesting both scientifically and therapeutically to now have this pathway that can be activated by this small molecule. And so one, from a scientific perspective, you say, okay, so what are some of the outcomes that we might see if this is a small molecule? And the first one is that this might be able to transfer intercellularly, so enter between two cells. And there are systems that allow molecules and metabolites to be transferred between cells. There are different ways to connect cytosol between cells, and one of those ways is through gap junctions. There was some thought, okay, perhaps in one cell that's infected by a DNA virus, cGAS becomes activated. It makes cGAMP. It could transfer this signal to the nearby cells and activate their type 1 interferons, activate their protective mechanisms to viruses without having to actually see that virus. And so that could be evolutionarily advantageous, because normally we would say cytokines would do that, but this is a much faster system than a cytokine. You don't have to wait for the transcription, the translation, the secretion. The molecule could just diffuse right to the nearby cells, activate, and like you started to create like a really strong antiviral response.
Yeah, go ahead, Matt.
No, I was going to just point out, like, this idea of spreading antiviral innate immunity is not something, novel to this, gap junction sort of thing. And you're pointing out, I think, a larger conversation that is generally understood, but just not at this speed. Right, so the type 1 interferon system in general, when it's discussed in intro immunology classes, is essentially exactly this system, which is that you get viral infection, you turn on a type 1 interferon cascade, those type 1 interferons get produced, right? They have a self-amplifying mechanism, essentially, so that cell itself starts ramping up its own interferon-stimulated genes, but also that interferon then goes to the surrounding cells, activates those receptors, and so this is how you sort of propagate an antiviral response locally. And everything that you're talking about is so cool because it takes time, even though we talk about that as this ridiculously fast innate immune propagation, that still takes, minutes to hours in order for that system to turn on effectively, right? Up to 24 hours for some of it. And what you're talking about is really just like literally shooting small molecules in all directions almost instantaneously.
Yeah, and the other piece is that it's dependent on transcription and translation and then excretion, right? So you have to get those proteins translated into the ER through all of the sort of extracellular processing pathway and then released out into the cytosol. And every step of that pathway is something that a virus could interfere with. And we know that there are viruses that interfere with many of those steps of the pathway. So having basically an enzyme that can just do a couple chemical reactions, generate small molecules, they don't require anything else to get over to the other cell. I think that's quite ingenious. And I remember so clearly, like, the idea of gap junctions with cGAMP hadn't occurred to me. But the moment that Kate described that idea to me, I was like, oh, yeah, that clearly must be a thing. Because it would be such an efficient and useful addition to this system. It seemed impossible that it wouldn't be true once she had brought it to my attention. And then it was shocking that she couldn't find it, basically. I don't know how much you actually tried, but I remember you sort of like looking at it and then like not seeing anything.
I did find it. I got scooped.
I believe we're all fighting now. Now we're in a fight.
So I did do early work on this with cGAMP and like found that it was like actually set up a system genetically like this one. And then found that it was sensitive to gap junctions, both the drugs and the knockout of the gap junction. But where I was going to start is then Veit Hornig and Andrea Blasser, they published that cGAMP can be transferred through gap junctions. And so they basically show the really similar. So they show something really similar to what we see in this paper here. Though they don't use a straight up genetic system in this way, but they show basically that like you can find that these molecules are transferred through gap junctions. But I think that where this landed was maybe like this is like a greater maybe this is a greater theme throughout all of innate immunity. Maybe there are many of these molecules that are made. But I think that through like mostly like bioinformatically assessing, all of the different types of proteins, it doesn't seem like there are a lot of these syntheses that are present. However, there is one system that's very analogous to this that is present, which is the OAS-RNaseL pathway, which is what this paper is looking at. OAS is structurally homologous to cGAS and is somewhat parallel to the cGAS-STING pathway in that it binds double-stranded RNA and it produces this small molecule called 2,5-oligoadenylate from ATP. And then this small molecule activates RNaseL. So it binds RNaseL, which is an endonuclease that cleaves both host and viral RNA. And these cleavage products can activate RIG-I signaling and thus interferon. So it's similar. So we have like this small molecule that can also activate this pathway. It can both activate an effector mechanism, so something that works very directly against viruses in the RNaseL activity, but then also can amplify some interferon signaling through these ligands that are produced, like activating this RNA sensing pathway. So this molecule that OAS makes, 2,5-oligoadenylate, it's a small molecule. It should be able to travel intracellularly to activate innate immune pathways in cytocellularly connected cells. So this is actually, I did try this experiment. I could not get this experiment to work.
With CGAMP or with CGAMP or with 2,5-A?
With 2,5-A. Okay. I think for that, so that's why I was so excited to see this, because I tried this exact assay. I think my perspective was like, if the assay comes along really fast, then I'll go down this road. But if the assay doesn't come along really fast, I'm just going to move on. Like, I'm not going to struggle over trying to like see this activity. But if the question is, can 2,5-oligoadenylate travel intracellularly, activate innate immunity? This paper has shown really nicely, yes. That's a spoiler. Yes. So that's sort of where we start. And I feel bad for, the RNA people, the OAS people to say, let me give you the background on this paper. It's DNA sensing.
Sure.
But I do think that I do think that the DNA sensing really highlighted, the mechanism of that really highlighted, okay, let's look at these molecules. Let's look at like different mechanisms of how these molecules actually might be promoting like an antiviral state, activating innate immunity.
Yeah, so Kate, how long has this OAS pathway been around? Because I don't remember. I remember very clearly CGAS and CGAMP. I don't remember this coming out, and so it must have been.
Oh, it's before.
It was before.
Yeah.
Oh, really?
Oh yeah. I think don't hold me to it. I think the early 2000s. Maybe it was described as it's written here in like 2003. I could be very wrong on that. To be perfectly honest, I did not go into the original papers because, again, I was just going to talk about the intro as like things that I know very well and then be like, it's just like this.
The 2,5-A system, modulation of viral and cellular processes through acceleration of RNA degradation in 1998. It's the first citation
close.
of this pathway.
Yeah, you're right there.
Of this
Thank
paper.
It's right there.
Pretty good.
Wow. Okay, so super old school, but I don't remember. Yeah. I don't remember this being a thing. Like we were talking about RIG-I and MDA5 and all of that stuff. Like,
yeah.
Why did this never come up in lab meetings, or like why did nobody talk about this in our lab? I don't remember any of those conversations.
I did work on it, Kevin. So
was it while I was there?
It's because you simply weren't paying attention, Kevin. You simply were not paying attention.
So I think maybe part of it is that some of the initial. I think that like within innate immunity, there's like a huge focus on activating type 1 interferons and type 3 interferons, and really looking at that initial very powerful path, like these very powerful pathways that are going to then turn around and activate adaptive immunity. And so like these molecules that are really important for instructing the type of response that you'll see from the adaptive side. I think
and this is intrinsic to cells because you're just degrading RNA locally.
Yeah, so like these intrinsic mechanisms, antiviral effector mechanisms, I think that these are studied more by people who are like virus host interaction type people versus like maybe someone who would call themselves like a dyed-in-the-wool immunologist. Okay. So I think that these are more like your virologists or like virus host people that focus on these pathways. But I think for a time there was a bit of controversy over how immunostimulatory the RNaseL ligand is, and like, is the main thrust of this pathway to activate type 1 interferons, or is it to cleave RNA? And I think in this paper they can put together both activities as being relevant for the pathway. But I do think that like with all the RIG-I MDA5 MAVs work, that's like really the focus on, and this pathway is like upstream of RIG-I and MAVs, but that pathway is like the focus of like DNA or RNA sensing.
I would like to submit a Freedom of Information Act request requesting all emails to and from Kevin Bonham with OAS in the body of the email, because I do not believe that Kevin was actually unaware of any of these things.
No, I mean, it is entirely possible that we had many deep conversations. I just have zero memory of them.
No. So when I say like, I had an idea that like because of the results from CGAMP that 2,5-A would also go through gap junctions. It would also stimulate this activity. I had a heck of a time trying to put together this assay to actually read out either the RNA cell cleavage or how they read out with interferon. I just couldn't get that to work. Yeah, so just for whatever reason, I just couldn't get it to work.
Let's get into that paper, because I felt like for this paper, the methods in particular were super impressive. I have like different highlighter colors that I use for like background versus methods versus like conclusions. And like my reading of this paper, it's just like littered with orange, which is what I use for the methods section, because there's so many creative and like pulling together different models, pulling together different inhibitors, pulling together different cell lines and like cross-species stuff. So like let's get into some of those methods, because I think they're not sort of traditional immunology the way that I would think about it, but very cool biochemistry and like really like neat mechanistic stuff. So should we get into those details?
Yeah, let's get into the details. Okay, so figure 1, panel A. So here, so let's start by talking about like what you're saying, the actual coal culture model here, and this is really the workhorse model that they use to show the activity throughout a couple of different figures. And so the way that they approach this is that they take their cells, and they either have cells that can make their ligand, or they have cells that can respond to the ligand. And in that way, when you coal culture the cells, if you do see the response to the ligand, you know that ligand had to have been transferred from one cell to the other cell. So specifically what that means for this paper is that they make cells that they knock out in RNaseL, so then they have all of their OAS enzymes, so they can produce 2,5 oligoadenylate in response to double-stranded RNA, but they cannot respond to it. So they don't have the RNaseL to actually cleave the products. And then in another cell, they knock out OAS 1, 2, and 3, so they can't make the 2,5 oligoadenylate, but they can respond to it if it does enter the cell. So they have competent RNaseL. And unfortunately, the way that they have to label these is OAS-123 knockout, that is going to be your responder cell, and then RNaseL knockout, that will be the producer cell.
Although I have to say,
So
the little icons that they keep a consistent color pattern throughout the paper, like where a particular model cell is like, used throughout the figure, those I found very helpful, and more people should do that kind of thing. Very good graphic design here. A+.
yeah. So they first just go take these cells in isolation or in a cold culture and transfect their producer cells with poly-IC. So
Intracellular
in the case
RNA mimic.
Yeah, exactly. And so when they have their cells that are knocked out in one of the two components of the pathway, they see no RNaseL activity. So that's what you would expect. The cells are either unable to make 2,5 oligodenylate, or they're unable to respond to it. But when you cold culture the cells, what they see is that in the presence of poly-IC, they do see RNaseL activity. So they look by bioanalyzer and they look at the RNA fragments that are present in the cell. And so they see the ribosomal fragments, the 28S, the 18S, that's going to be the most dominant RNA that you find in the cell. And then they also see, if you look at the figure, like these little lines, and those are all your cleavage products, and they are not present in your negative controls or in your single culture cells. So the second thing that they do in this figure is they then say, okay, how is the 2,5 oligodenylate getting from the producer cell to the cell that can respond? And there are a couple of ways that this could happen. It doesn't have to happen through gap junctions. It could be happening through extracellular vesicles. There could be, these cells could be lysing, and you have like just these metabolites out into the serum.
And that would make sense too, because
into the culture.
if viruses are killing cells, if those cells are either dying because the virus is lytic in some way or through apoptosis, like it would make sense that there might be some other mechanism to get that stuff.
Which is how ATP works, right? In a setting like this, where you have apoptotic cells that release ATP that shouldn't be out there, out into the outside world, and then other cells pick that up, right? This could be a version of that.
Exactly. Yeah. But what they do is they treat with a gap junction inhibitor. I think this one is carbinoxolone.
CBX.
Probably somewhere. Yeah, they call it CBX. And they find that in the presence of both poly-IC and CBX, there is no more RNA cell fragments that are made. So the responder cell is not getting the oligodenylate. So suggesting that the oligodenylate is dependent on the functional gap junction to transfer between the cells. They then, using the same assay, the RNaseL cleavage assay, I really like this in that it's like a direct readout of activity. The rest, they do another assay that reads out direct RNaseL activity, but they then switch over to a transcriptional assay. But I really prefer this assay.
Yeah, well, they say somewhere that the transcriptional assay is like more sensitive. This is a little bit later on, but I agree with you that this, like, showing the direct activity is very clear, and like those gels are beautiful. Also, I don't know. Even if they give that PTSD.
The sensitivity makes sense to me, but I also think it's a lot easier to run.
Sure, fair.
Yeah, no, qPCR is way easier than running a good RNA or protein gel, right?
I think this is on a bioanalyzer though, yeah. So like it's more so, but yeah. So then in 1B, they're saying, okay, let's step away from the drugs. Also, those gap junction inhibitor drugs, they are very finicky. They make the cells pretty sick if you keep them on for very long. So, they confirm the the action of that drug actually blocking gap junctions through these CRISPR knockouts of connexins, so connexin 43 and 45. So connexins are the proteins that make up a gap junction. So they form these channels so they form like these pores in the cell, and then you have like these hemi channels, and then they can basically form on either side of the two cells that are connecting. So you get these connected cells. And so connexin 43 and 45 are basically the most common connexins that you'll find that are like the most ubiquitously expressed. These cells probably express a fair number of connexins. There's some like very specialized ones and lowly expressed ones, but these are the main ones. And something that was shown in the CGAMP gap junction paper was that, and this is why we thought it was diffusion, they find that it's not like one specific connexin that's responsible for transmitting CGAMP from cell to cell. It's just whatever connexin is forming gap junctions, CGAMP will flow through it.
So do you know, I mean, I know that there is some, there's some amount of just passive diffusion. There's some things that sort of need to be actively moved in. Is there also like exclusion? Like, is it any molecule below a certain size is going to diffuse, or is it like how specific is the passage? Like sodium ions or whatever are going to transfer, and nucleotides are going to transfer, but like how permissive is that? Because I can imagine also like this is the kind of thing that a pathogen would exploit if it could, because if you don't have to leave the cell to get to a neighboring cell, it's much easier, like pathogenesis wise, right? And there are some bacteria that do this, where they'll like, is it Listeria that makes little rockets that like shoots, just like pushes itself through barriers? There's one of those bugs that does that.
Yep. So my understanding, not as someone who studies gap junctions, is that they are just how you're saying, as long as you're a certain size or like your charge doesn't exclude you, there is a passive diffusion that occurs, and that this is not like an active
size.
Up to some size. And actually, when I was looking at this originally, I believe that at the time I thought 2,5 oligodenylate might be too big, and I don't remember. I mean, that was a thought that I had, was like, I remember I was talking to someone who studied gap junctions, and they thought that it was too big to go through the gap junction. But clearly not.
Bad advice. Bad advice.
But
although the fact that they can just inhibit these particular connexins, I mean, maybe there is some active movement, because they can also sort of shuttle things that would otherwise be excluded, right?
I think that in this case, these are just like very, very widely expressed.
Okay.
And my guess would be that they are responsible for the vast majority of gap junction formation. And so, maybe it's 80%, and that 20%, you're just not going to see it on the gel.
Got it.
It's, yeah. So they show genetically as well that if you knock down the building blocks of gap junctions, you will also prevent the responder cell and the producer cell from transferring that molecule. In the next couple of panels on this figure, they introduce their interferon assay. So essentially what they're saying is they switch from delivering a ligand to activate OAS to using a constitutively active OAS, and then they culture these human cells, importantly, that are expressing OAS with mouse cells. And so now they're saying, okay, if our producer human cell is making OAS, is making 2,5 oligoadenylate, our recipient mouse cell will be able to receive it and it'll activate RNaseL, which will activate RIG-I MAVs and turn on both type 1 interferons and interferon-stimulated genes. And so they look at IFIT, CXCL10, classic genes. And so in this case, again, what they're seeing is when they cold culture these cells, when they cold culture the cells with increasing amounts of their donor cell, they get more activity in the MEF. So they're getting, as they increase the amount of cells that overexpress this OAS, they get increased interferon made, they get increased interferon-stimulated genes made. And then they cold cultured these cells in the presence of, again, this gap junction inhibitor, and this transcriptional response goes away. The one piece that I really liked was this IF that they do. So I think one of the immunofluorescence that they do, yeah. So one piece that you just like really want to understand is that, you know, only your cells that are like your specific responder cells are the ones that are making that interferon, and it helps, you know, validate some of like the ticky-tacky pieces of your assay, but also just gives you really pretty pictures to convince you of like the polarity of signaling and like what is actually responding and what is not. And so they basically take this cold culture system and then look at it underneath a microscope, where they label their cells that have the OAS overexpression. They then take their recipient MEFs. I guess I didn't say what this was, but this is a mouse embryonic fibroblast. So it's just like a very widely used line for mouse studies. Then they look at their cold culture, and what they do is they stain for interferon-stimulated genes, so IFIDM3, and they see that activation is occurring in the presence of your producer cell. The activation occurs within the MEF, and that if you cold culture the cell with increasing amounts of this gap junction inhibitor, that activation in the MEF goes away. So basically, you see like bright green cells when the OAS overexpressing cell and the recipient MEF are co-cultured, and then it starts to dim as you increase your gap junction inhibitor. So just like a really nice picture to show that, you know, these aren't artifacts of cold culture or artifacts of qPCR.
Yeah, and it's super clear because you can just see the differently labeled cells, and only the cells that are the recipient cells are lighting up with the interferon, stimulated gene. It's quite lovely.
I will say the only ticky-tacky thing I had was when you do transduce these cells to overexpress, you know, to overexpress a protein, you have to use some sort of like you have to introduce some sort of like stable DNA. That stable DNA can activate CGAS, which then can activate CGAMP, which itself will be transferred through gap junctions to activate interferon. I assume that A549s don't have competent CGAS in them, but that would be one piece to like control for as you go through these experiments, is to make sure that you're not. And I would think that you most likely are juicing the cells to respond a little bit better to the 2,5 oligodenolate. Not that I think that this would only be CGAMP. I'm not saying that. But it is just one thing to control for, because they don't actually have a really convincing. Maybe it's in the IF. They have one panel, but in the transcriptional, it would just be nice, because I don't think they have a cold culture of a donor that is the control donor. It looks like all the cold culture is with the overexpression. Anyway, that's a small thing.
You've got to juice the cells to maximize the gains. That's what you're saying.
I would not. Well, like, you know,
Matt's just being dumb. You don't need to respond to that, Kate.
No. I think that a lot of innate immune, I think that it's very difficult to have pure ligand So in the case of like intracellular DNA, the DNA that you would like transfect into cells is oftentimes highly contaminated with LPS. And so I think a lot of these studies that we do, the ligands are not pure. So and when they are super, super pure, they don't respond as well. And I think that there's probably is a little bit of maybe some like priming of the pump with like small amounts of like other ligands. Not to say I want to be like very clear. I don't think that that's a bad thing. I think it probably just helps with the phenotype to study study what you are working on.
Like they are using
You
extensive
use controls.
to show that is not
Exactly.
the
Yeah.
cause. I'm just mindful of time. I know that Kate has a heart out here, and we're only through figure 1. So I'm wondering if we should sort of jump through and sort of summarize a couple of figures so we can get to the end. So like figure 2, they're basically showing that it's not only this 2,5A isn't only going through gap junctions. It can come in from the exterior of the cell as well.
But only sometimes.
In both mouse and human, right? Only sometimes. And it's inhibited by serum, which is kind of interesting. So they show that they can like add different serum and it causes different effects. And then, honestly, I can't remember what...
And I want to... Real quick before we move on, I want to point out that what they do there is they basically test 7 different lines, right? So if you did this experiment in one of those 7 lines, you may not actually know the answer, right? There's a really good reason that we study diverse models, right? We don't just rely on output from one, for example,
heterogeneous,
cell line,
not diverse, Matt.
No,
we want to keep our
I'm going to use diverse as many times as I damn well please. Thank you. It's just really important to point out that like if you were really narrow-minded about this and you just selected something and you went with the answer, you would not actually have the answer. Some cells seem to be able to pull this signal in and respond to it. Other cells just sort of leave it in the extracellular space where it sits there and rots, presumably.
Right.
Exactly. Like, so if you were to have used 293Ts instead of the A549s, you probably wouldn't have seen the result in the coal culture because the 293s are able to, like, if there was any of it available for them to import, they would have been able to. Of course, I will say the import is pretty sensitive to, it seems like, degradation. The molecule is pretty sensitive to degradation, and there's like this, I think like the terminal triphosphate is necessary to activate RNaseL, which is like very easily degraded. But yeah, I think you like bring up a really good point. And maybe this figure came from, someone putting together some of these coal cultures and other cell lines and saying like, why is there some difference between the different cell lines?
I feel like this experiment came first. They were like trying a bunch of stuff and they noticed like, oh, it's only working in some. Let's go look at the ones that it's working in. But
yeah, agreed.
Yeah, and then to like tie that to an activity, like a difference in activity, and actually prove that out is very, very shrewd thinking.
Yeah, totally. So yeah, figure 3, they're just showing that it's exported, right? They're using some fret, which is this cool microscopy technique where like you see when things get close together, you get a different fluorescent signal.
This is actually a slightly different setup. It still is fret, but this is this RNaseL direct activity assay where they have a ligand and there's a fluorophore and a quencher.
Right.
And so if the ligand has not been cleaved, you don't see any signal, but once RNaseL is activated, it cleaves. The distance now
releases
between like the quench and the fluorophore releases, and you can like now see the signal. Right. So this is a very cute assay. I like this assay. This would have been my workhorse if I were doing this.
Yeah, it's cool, and it's a very clear phenotype too, and you can get like nice time resolution
really clear
on it, which is cool.
Fluorescence is great, and that's why I don't do gels anymore.
So I sort of think that figure 4 is another like the gap junction piece to me is like really neat, but it feels like they layered like several conclusions into this paper, like each one of which is really well done. But like figure 4, they're showing a specific transporter that is actually the thing that is responsible for pumping the 25A out into extracellular space, which is kind of cool.
Yeah. Yeah, so figure 3, they show that it can be exported, and then figure 4, they tell you what actually was doing that exporting. This, I will say, my one question with figure 4, so they identify these ATP binding cassette transporters as, they say, you know, there's a lot of different exporters and importers of CGAMP. Let's kind of look at the CGAMP ones and see if there's some overlap between 25A, and they do see that, like, in this family of exporters, you know, if they use like this drug that kind of hits all of them, they see less export. I am curious, like, how did they know? I actually thought maybe this was the first figure of the paper, actually. Like, this is where it started. Because I was like, how did they know? What led them to this one in particular? Obviously, I assume they tested a lot of things that didn't work. Maybe they tested every known exporter. Maybe they did a screen. Maybe they had the extracellular assay, and then they started screening for the exporter. And then sometimes you just say, this is what it was, because here's the activity. So I am curious, like, what led them to this path, but yeah, this is they do see this nice response from the inhibitor, and then they can just turn around, knock out every single exporter or transporter that is expressed in these cells. They do a cute little comparative, right? So they say, here are the cells that can export 25A out of the cell. What are the ATP binding cassette transporters that they share? And then those are the ones that they knock down. And they find one that's probably responsible for like 80% of the export. Maybe like 75. But
well, they're doing knockdown, so it could also just be that their knockdown isn't like the most efficient, right?
It looks really good.
It's great, yeah, but it's like unclear if that's if there are other things that can export it, or if it's just like residual.
We live in a post-gel world, and if there's nothing on that gel, there could still be something in that cell. Oh no, that rhymed.
She wrote that ahead of this episode.
These people are.
She knew that she was going to bring that out.
These people are very good molecular biologists and biochemists. That's just like the take home from this whole paper is that these people have good hands.
Yeah, they're good at this.
Yeah. And so they do the knockout of their transporter, and if they do, they see that the intracellular levels stay high. They see, which is actually kind of interesting, but the extracellular levels plummet. Sort of interesting, actually, about the intracellular staying high, but the extracellular dropping down, that there isn't like this like cognate increase. And I wonder if it's just a signal, like the signal between the amount of extracellular 25A is so low that you wouldn't really see it as like dropping out of the intracellular, or maybe they're at the max of the assay.
Yeah,
like,
I was thinking it was like you're at some like limit of detection, upper limit of detection, and it's just like, and those are log plots? No, they're not log plots.
No.
Well, still.
But yeah, so I thought that was interesting, but there could also be like some sort of feedback within the cell that at a certain amount of intracellular ligand, you auto inhibit your enzyme to like as like a shut off.
Pathway. Right.
Yeah, especially if you're a bacteria. Okay, so the next. How are we are? Okay, there's 3 more figures. Okay.
Yeah.
I like this next figure. So, okay, here's like the tricky thing. You show that this molecule passes between the cells. Great. You show that it activates, interferon, it activates RNaseL when the 25A was produced in another cell. Great. But ultimately you want to show that the action of this molecule being passed when it goes into the recipient cell, it actually does induce a demonstrable antiviral effect. Because if you show that the molecule passes but it doesn't have any real activity, big whoop, who cares? A lot of things might be happening in a cell that you can observe that maybe you can't really tie a biological result to or outcome to. So in this case, they start to look first at infection and then at tumor growth. And so the infection I really liked. Let's see here. This one is, okay. My notes to myself. Jeez. Kate. Past Kate was like, I'm on it. Future Kate is like, okay, this is literally my note. Love this. Very simple.
And now you can't remember what's actually going on.
Past Kate, what are you doing? Okay. So that is an all-time classic note to myself. I always leave notes to myself like that in papers. Okay. So they take their knockouts, so their RNaseL or their OAS knockouts, just like they did in the coal culture experiment, and they infect these cells with VSV, and then they coal culture them with wild type cells. And so in the RNaseL knockout cell, that cell should be able to make 2,5A. And in the OAS knockout cell, none of that will be made. So essentially what they're saying is when VSV infects a cell, 2,5A will be made. If we then coal culture these pre-infected cells with wild type, the wild type cells will get infected from the cells that are infected, but they also should be able to receive 2,5A upon that coal culture. Okay. And so if the cells that can make 2,5A have less viral replication in the coal culture, that means that the 2,5A that they made was antiviral because they can't respond to it, right? So if there's any 2,5A additional suppression of viral infection, it came from that wild type cell receiving it. And so this is exactly what they find. So in the cells that are competent for the enzyme, they see that there's less viral replication than in the coal culture with cells that do not have the enzyme. Interestingly, the wild type cell will have the entire pathway. So it's really looking at that it's really looking at that kind of preventative shot of oligoadenylate. And then in the presence of gap junctions inhibitors, the cells that do have ligand to transfer, they do not suppress quite as well. But in the cells that don't have any ligand in them, any extra 2,5 oligoadenylate, the gap junction inhibitor does nothing to the viral infection, which is a really great control as well, because again, those gap junctions, you would think maybe they might have some effect on viral replication, maybe they just make the cells a little sicker. But we see that basically the infection is the same. You guys also thought that was very simple. Love this, right?
Oh yeah, absolutely, so simple.
No, I mean this is where in the paper I was like, this is something I recognize. We're talking about cells getting infected by a virus and preventing other cells from getting infected by that same virus through dissemination of this thing. So no, I think it actually is a simple assay, and I think it speaks to exactly what we were talking about at the top, which is the spread of this antiviral state rapidly cell to cell without the need for transcription, translation, and the downstream signaling.
Yeah, simple. I think simple conceptually, but again, like with a lot of this paper, the density of method here, right? We're talking about different cell lines with different proteins knocked out. Co-culture, you've got to make sure you're mixing the right cells with one another. You've got inhibitors, you've got like the viral stimulation, you've got artificial stimulation. Like it's a very complicated and like when you have underneath your bar chart, when you have like 6 things that you have like plus and minus combinations with, like it's complicated. Ultimately, the result is straightforward, I think. But
yeah,
I think it's methodologically very sophisticated and
I would say
hard to pull
elegant.
out together.
Elegant?
Yeah, I like that.
Yeah. All right.
So the second one, really similar idea. Now, instead of pre-treating the cells, or instead of infecting the knockout cells, because you could say maybe the knockout, is more or less resistant to VSV. Let's look at this a different way. They transfect the cells with poly-IC or MOC, so that poly-IC, what we were saying before, activates OAS. So if you have OAS in the cell, you will make 2,5-oligoadenylate. But if you are knocked out for OAS, you will not make that molecule. They wash the poly-IC off and then co-culture the cells with wild-type cells. So this allows for these cells that have OAS to transfer their 2,5-oligoadenylate, and then they infect the co-culture with VSV. And again, they show that if they do this pre-treatment with poly-IC in the RNaseL knockout, which has OAS, you can suppress viral replication. But if you do the pre-treatment in cells that do not have OAS, the suppression relative to no pre-treatment is less. So there actually is some suppression. It's probably from some sort of like interferon, but you don't see. But the suppression is much more dramatic when you have the functional OAS. And then this last bit, they're looking at the role of the exporter during infection. And so they infect wild type cells or their transporter knockout MC38 cells with VSV GFP, and then they measured the replication after 24 hours. And they show that their knockout cells show significantly higher replication, and this is suggestive that in the knockout cells where there is not any of that export occurring between cells, that the virus is able to replicate more effectively. So essentially, like that transit, but even though the cells all can make 2,5-oligoadenylate, the fact that they're not getting a little taste of it before the virus comes, they're less able to control the virus. Yeah. And then the other side of the paper, this is just looking at a different method to actually quantitate the 2,5-oligoadenylate in these experiments, and they see that the amount that's being made across these different designs is like very similar. So it's a nice control just for
it's not like the virus or whatever stimulation is blocking the production of the molecule.
Yeah.
It's more evidence that the mechanism that they are talking about of the moving of this molecule between cells, whether through gap junctions or through export, is the thing that is different and causing different phenotypes. Yeah.
So
lovely.
If this were me, I'd stop the paper right
100%.
there. I'd say, we got it.
My note.
It's antiviral.
My note on
yeah, we did it.
My note on figure 6 is, this is just excessive. That's.
It's like, because I do think that for me, for my interest, it's just like, was it antiviral? And I feel They showed 3 different cultures. They have these really nice controls. It's antiviral. Yeah. Great. But this is where we sort of like now start to veer into, I guess we're going to talk about cancer vaccines.
Yeah, but it's,
and we'll have to.
In immunity paper, so you can't get away with just 5 super dense and methodologically complex experiments. You gotta You got to do more.
Right.
Yeah. But so I will say, because I think maybe I'll just explain the vibes of 6, and then maybe we can pick up on cancer vaccines in a subsequent episode. But essentially what they're trying to show is this pathway functional in vivo, and is it affecting like cancer growth? So a lot of these pathways that, also restrict viruses have roles in restricting tumor growth. And so they use a model where they're taking these MC38 cells to grow tumors in mice. And, they've already done some characterization of these cells throughout the paper. But they're essentially just looking at, okay, let's look at the contribution of OAS in these tumor cells in tumor growth. And they say, okay, if you don't have OAS, the tumor grows more. And then they look at, okay, what's the contribution of the exporter? Is the exporter important for controlling tumor growth? And they say, yes, if you don't have the exporter, the tumor grows more. In the next couple of experiments, they're saying, okay, is it important? And this is actually where it gets interesting. Is it important for the mouse where the tumor is growing? So not the tumor itself, but the mouse in which the tumor is growing. Is it important for that mouse to have RNaseL, which is the enzyme that's activated by the 2,5 oligoadenylate? And the answer there is also yes. The mouse needs to have RNaseL, which suggests that some cell of the mouse is receiving 2,5 oligoadenylate to control
cancer.
To
be clear,
to control the tumor.
We're transporting a tumor from one mouse, or we're taking tumor cells, and we're putting them into these mice with different genetic backgrounds. So the tumor itself, activity within the tumor cells is the same throughout all of these experiments, right?
Yep.
But so it's the recipient mouse, their cells need to have the enzyme that responds to the 2,5A, even though there's no difference in the tumor cells themselves.
Exactly. So it's not that the tumor cells are transporting 2,5A to each other,
importing it.
To be honest,
yeah,
or importing it. That to control this to control this growth, the mouse has to respond to the 2,5A, and they show that in this next panel where they say, okay, let's knock out the exporter. So if we have a wild type or knockout exporter tumor, and then we put it in the RNaseL mouse, is there any difference? Meaning, if the export was important for the tumor, and like the RNaseL and like the mouse responding didn't matter as much, is there any difference in tumor growth? And they say like there's no difference here. They also finally do my CGAS knockout control. But so essentially where I think this gets interesting is that they look at RAG1 knockout mice and IFNAR1 knockout mice, and they put the tumor into those mice. And they then...
so just RAG1 knockout, no T cells, no B cells. IFNAR knockout, no interferon receptors. So you can't respond to the interferon that is released.
Type 1 interferon.
Type 1
yes.
interferon. Yeah, thank you.
Yep.
And then they treat the mice with either 2,5A or 2,5A complexed with lipofectamine, which should like deliver the molecule to the system, and they see that there's no difference in the control of tumor growth if that mouse does not have T cells or B cells or does not have type 1 interferon signaling. So I think that part is really interesting because what they've really built is that this system is important for the control of tumor growth, but specifically, it seems like in the immune compartment of the recipient mouse.
Which is
yeah.
I would not have predicted this result. from the rest of the paper. I would not have predicted this result at all.
Yes. And so, like, this is the point that I want to end on. So when CGAMP was found and we saw that it was a small molecule, we also thought, hey, maybe treating tumor cells with CGAMP and activating innate immunity and trying to create these tumors that can be recognized by the immune system, maybe this is going to be really therapeutically beneficial. And in the mouse, it was. CGAMP acts on the tumor cells to activate STING. It turns on interferon, and the tumors melt in the mouse. However, in the human, it's absolute trash. It does not. I mean, it has been. Maybe I spoke too harshly. It has been a slog. It has been riddled with clinical trial failures. Like trying it in some like dual therapies, monotherapies. The companies that started down these roads, like it has not worked out well. It has not been, and there's been some interesting papers to say, okay, why might this be? One of the things I noticed when I was working on STING was that a lot of cancer cell lines don't express STING. A lot of cancer cell lines have chromatin instability, so they have DNA coming into their cytosol. They might be overactivating STING, and so the STING activation might be a feature of tumors, or at least some tumors, leading to, this could lead to, like, we could say, like, maybe there's a resistance to the STING signaling, maybe, and I think a lot of pathways have this. When you activate, there's some compensatory mechanisms to turn off or make the cell insensitive to it. So I think that there are just a lot of there's a lot of different ways that like a tumor cell might be regulating this pathway. But in this case, they're saying it's not the tumor cell that's responding it's not the tumor cell that's responding. It's not that we add in 2,5A and the tumor shrinks because we activated some immune response. It's that we add the 2,5A and it activates the T cells and the B cells, and then those cells theoretically are what are controlling the tumor volume and the tumor growth. So it's sort of like saying, okay, trying to create this activated tumor environment, it has not worked, even though, this was such a great idea and like such a great way to utilize like biology that we find. But in this case, they're saying, okay, we're not going to go at the tumor, we're going to go at the immune system. And so that's sort of where we're like, cancer vaccines are back, baby.
It's a B-cell.
Yeah. So, I think, we're going to start all over again. We're going to just repeat the cycle of all of the STING agonism.
Also, Kate will be looking for venture capitalist funding around this idea. Yep. Be prepared.
Yeah, this is a very cool paper.
See, I told you we'd get to cancer vaccines eventually. You were nervous initially.
It's great. It was a very intense paper to read, just like constantly needing to refer back to like, what was this inhibitor? What was this? But like, again, like the graphic design, I have a few quibbles with some of the figures, but I'm not even going to talk about them because they're minor compared to like having those diagrams, having those icons in the figures. So helpful, like really helped clarify, and it's a complicated paper, so it was really necessary. And so kudos to these folks. I realized as we were halfway through, I just wanted to correct. There's actually two corresponding authors. So Jianjun Wu is the second to last author, is listed as co-corresponding. Although it seems like there's not co-first author, so maybe this is like a joint trainee or something. I do find it funny that one of the co-corresponding authors is listed as the lead contact. So like what does co-corresponding even mean under those circumstances? But leave that aside.
Yeah, you still. Yeah. Having done this a couple times, like one person is still responsible for going back and forth between the editor, and realistically, you're sharing it, but that's an asterisk that's added to the actual publication.
Yeah, it's an asterisk.
That's
on
not
the
to say
asterisk
that these
of-
people. Yeah, that does not mean to say that they did not contribute equally. It's just simply logistics. The definition of a corresponding author is the one that talks to the editor, and there can be only one.
Yeah,
Like Islander.
fair enough. That is a very dated reference, Matt. I think that very few people will actually understand what you're talking about, but that's fine.
Agreed. Agreed. Dated is what I do.
Is there anything else that we wanted to talk about on this?
I don't think
All
so.
right. Well,
We're good. I
in
think
that
Kate
case,
needs to get the hell out of here.
this has been Audio Immunity. I'm Kevin Bonham with Matt Woodruff and Kate Franz. You can and you should support us on Patreon at patreon.com/audioimmunity. We are also at audioimmunity.org, AUDIOMMUNITY.org. You can find links to all of our past episodes. You can find other ways to support us. We would really love comments on Patreon about, we have a couple new subscribers since our last episode, so thank you. I didn't ask anyone if we could read their names off. Maybe we should. But go to some of our websites. Get in touch. I'm on Blue Sky. Matt's on Blue Sky. Kate is probably lurking on Blue Sky, but won't give us her handle. Find us. Come watch our old episodes. They're still good. And the music at the beginning and end of the show is composed by Rachel Reinick. And we'll see you next time.
See y'all.
Yay!
Sometimes you get lucky. Sometimes you get lost.
I was like, I don't know what you're talking about. It's like just really simple cognate cold culture of knockouts,
Yeah,
and then
it's
you just
cell
look for
culture,
the
man.
signal in one, and then also you take the lysate, and then you run your bioassay. What is difficult to understand?
Yep. No, we are down a rabbit hole of cellular and molecular biology here that I have left behind almost entirely.
I mean,
it's gone.
I have also left all of this behind, but it did feel like coming home. It did not feel like a PTSD.
It felt kind of like stepping over a cliff. Like, what am I doing?